Hi guys, welcome back. Today we're going to do a quick mechanism of the pathogenesis of celiac disease. So celiac disease is a bit of a weird condition. It's not really an autoimmune disease. It's not really kind of an infection or anything like that. It probably falls best into the category of a type 4 hypersensitivity, also known as a delayed type hypersensitivity, with some elements of an autoimmune response. However, the antigen that's being targeted is not a self-antigen, it is still a foreign antigen. So to start off with, so celiac disease, it's almost like an allergic reaction to this thing called gluten. So we start off with the ingestion of gluten. Now, gluten comes in various forms. It's present in things like wheat, rye, and barley. There are also some types in oats. So gluten is like, it's probably best defined as a class of proteins rather than a single protein itself. The main one that we focus on is gliadin. This is a specific type of gluten that seems to elicit the most potent response in celiac disease. What's important about gluten and gliadin in particular is it has a high proline density. And if you remember back to your biochemistry, proline is this special amino acid that it forms kinks in amino acid chains because it doesn't follow the standard kind of transformation. It kind of results in something more of a cis conformation in the protein. So it forms these kinks and things like that. And what that does is it actually confers resistance to the proteases in your gut. So it confers resistance to proteases. What that means is that the gliadin, instead of being broken down by your pepsin and your tryptase and things like that, it can actually make it through your gut, and those intact gliadin peptides can be absorbed by the small intestine. So intact peptides absorbed by small intestine. From here, two things happen. There's an adaptive immune response, which I'm just going to abbreviate to AIR here, so adaptive immune response. And then there is an innate immune response. So we're going to focus on those two things separately. However, towards the end of the mechanism, they do start to tie in together. So let's start off with the adaptive immune response. And I'll keep it the same color. So what happens here is there's an enzyme in enterocytes and in the gut wall called tissue transglutaminase. It's a bit of a mouthful, so we usually just call it TTG. So the small t is for tissue, then the TG transglutaminase. What it does is it deamidates. So this is important. It's not a deamination as normally happens when we talk about biochemistry. We talk about something being deaminated. This is deamidated. And what that does is it deamidates the glutamine that's in that gliadin protein to glutamic acid. This glutamic acid, under physiological conditions, it's negatively charged. So it's not a carboxylate group, carboxylic acid. It becomes a carboxylate kind of thing, deprotonated. So it's got that negative charge. So negatively charged. That negative charge is really important because that negative charge can then bind to HLA DQ2 and DQ8. So negative charge allows binding to HLA, DQ2, and DQ8. So HLA stands for human leukocyte antigen. It's also known as MHC. So MHC is kind of like a broad term for the protein in all species. HLA is the human-specific MHC. DQ2 and DQ8 are two alleles of this particular type of HLA. So it's DQ, which means it's MHC2. So you have HLA A, B, and C, and then you have dq, dr, ds, I think, or maybe dp, dq. Something like that. Anyway, most important thing is, sorry, I'm doing this quite late on a Saturday night, but this is MHC1 and this is MHC2. I believe I'm going to get that. I'm pretty sure it's DP, DQ, DR. So DQ2 and DQ8, they're both isoforms and alleles of this MHC2 class MHC class 2 molecule. Okay, MHC2 is important because MHC2 is what allows us to present to CD4 positive T cells. So our antigen presenting cells, APCs, present the gliadin proteins to CD4 T cells. It's important that these ones are CD4 because we're talking about a MHC2 molecule here. If it was an MHC1 molecule, it would be presenting to a CD8 T cell. Okay. From here, we split again. So, first of all, CD4 positive T cells proliferate. Cells proliferate. And they then provide T cell help to B cell. T cell help to B cells. And that results in deaminated gliadin. Here we go. This is a bit of a mouthful. Deaminated gliadin, IgG, and tissue transglutaminase IgA. Okay, so these are important. The reason these are important, they're not pathogenic, but they are used diagnostically. So when we're testing someone for celiac disease, we can do a non-invasive test, just a blood screen, looking for deaminated gliadin IgG, which is because you get this T cell help. And often what happens is this tissue transglutaminase, it's often taken up into B cells complexed with gliadin. And so because of that, B cells that are taking up both the gliadin and the tissue transglutaminase means that, let me draw this out. So we have this B cell here with its B cell receptor, and this, let's say that this is gliadin, and come on, blue, and this is the TTG. Let's say that this is a gliadin specific B cell. It's going to bind to the red part. It's going to internalize both, but the main thing that's going to happen is it's going to put gliadin on the... Gliadin is going to... Oh my goodness, come on. Sorry, my computer's running a little slow today. It's going to be expressed on the MHC2. Sorry, let's make that red. How about... Oh, wait, let me pin this. Okay. It's presented on MHC2 here. T cell comes up, activates the B cell. So even though there's production here... Okay, let me draw this out. All right. So, B cell, specific for gliadin, endocytoses both because the tissue transglutaminase is kind of complex with the gliadin. It presents both gliadin peptides and the tissue transglutaminase peptides on its cell surface. However, T cells don't recognize self molecules, so the T cell is going to recognize this and provide the help. And then we're going to get from here, we'll get all these antibodies to gliadin produced. So that's why we get the IgG. However, what we're also going to get is, if I draw another cell out over here, say now that this T cell is now actually specific to the TTG. So, our B cell. So B cells don't have as much primary. There's much more. There are many more B cells that have self-selectivity than there are T cells. B cells very much rely on the periphery and the T cells in the periphery to control autoimmune reactions. The reason for this is that there's a lot of bacterial capsules present antigens which overlap with our own tissues, and so we need B cells that are a little bit self-reactive in order to be able to fight off more infections. So, and usually what happens is that T cells, which have a much more rigorous selective process, they are released into the periphery but with no self-selectivity. They're very, very specific just for non-self antigens. And so B cells that are responding to self-antigen aren't going to be able to isotype switch and release a lot of antibodies under normal conditions. Okay. I'm getting a little bit sidetracked with this video, so I'll try and keep this bit brief. But basically now what's happening is this B cell is reacting to the tissue transglutaminase complex, the gliadin, uptakes them both. Same thing here is it's going to express both the gliadin and the TTG on its surface. And now what's going to happen is that T cells responsive to the gliadin are going to activate this B cell, despite the fact that its B cell receptor selectivity is for the self-antigen. And so now we're going to get release of these IgA molecules to the TTG. I might make another video about this at another point to try and make this a little bit more clear, because I know it's a little bit confusing and I'm a little bit tired at the moment, so I'm probably not explaining it very well. So I might leave that there for now and come back to that another day. Now, going back up then to antigen-presenting cells presenting gliadin to the CD4 T cell. So we've got the proliferation and things like that. What we then get is differentiation into a T helper 1 phenotype. Differentiation. Th1 cells, they can give, so the main thing that they release is interferon gamma. And then this is where we start to tie in a little bit with our innate immune system response. So we'll go back to that now. So right back up to here, your innate immune response. Having that whole gliadin coming through into the lamina propria, it's not normal, and having that much protein there actually causes enterocyte stress. Enterocyte stress does two things. Firstly, it upregulates this molecule called MIC-A on the surface. So MIC-A is a cell stress molecule, and that's going to become really important in a second. The other thing that these enterocytes do is they release interleukin-15. Interleukin-15 stimulates intraepithelial lymphocytes to accumulate and to adopt a natural killer cell phenotype. So that means that they upregulate the NKG2. I always get this wrong. So NKG2D receptor. So intraepithelial lymphocytes are CD8 positive non-conventional T cells. They're kind of like memory cells. They sit within the enterocyte border and kind of sample and make sure that things don't get infected. What they can do is that when stimulated by interleukin-15, they start to upregulate this NKG2D receptor. This is a receptor that we usually find on natural killer cells. So that's why we say that these start to adopt this natural killer phenotype. And just like natural killer cells, these cells are now able to go and kill cells that are going to activate that NKG2D receptor. Now, one of the things that does do that is MIC-A. So the recognition of MIC-A, so NKG2D recognizes MIC-A, and from that you're going to get immune destruction of enterocytes. Great. Other things that also contributing to this, so this interferon gamma release that we talked about before also ties into this and also helps upregulate that natural killer cell phenotype. What happens from here now is that we start to see some of the, We'll now talk a little bit about what we see in celiac disease. So some of the symptoms, some of the outcomes, what we see on histology, all that kind of thing. So first of all, the differentiation to T helper 1 cells combined with this chronic inflammation, this interferon gamma, means that people with celiac disease have a high incidence or an increased incidence of enteropathy associated T cell lymphoma, which is a type of cancer. The increase, this activation process here results in intraepithelial lymphocytosis. And then what we see on histology, because of this immune destruction, we see crypt hyperplasia and hypertrophy in an attempt to replace these destroyed enterocytes. So the crypts are where all the stem cells for the intestinal epithelial lining are, and so they start to proliferate more and more and more in order to try and overcome the loss that we're seeing from all these enterocyte death. So we see crypt hypertrophy and hyplasia. We see villus blunting. So instead of, so usually this is kind of what this is what the gut epithelium kind of looks like. So you have your villi here, your crypts here. This is kind of like, I guess, the middle layer. What we start to see in celiac disease is we get blunting of the villi and then crypt hyperplasia and hypertrophy. So we lose all the surface area from our villi because they've all been destroyed, and then our crypts are proliferating in order to try and replace the cells. So villus atrophy and blunting means that we then get, before we go into that, one final thing, this crypt hyperplasia and hypertrophy combined with the interferon gamma also results in an increased incidence of another cancer, which is small intestinal adenocarcinoma. Cool. All right, so the villus blunting ties into that crypt hyperplasia. Cool. Now we're going to go on to some of the symptoms that we see. Most of the symptoms that we see are due to absorptive deficiencies. Now, someone with celiac disease can present with a wide variety of symptoms, depending on how long they've been eating gluten for, how severe their reaction is, all those kinds of things. So we're just going to talk about all the different nutrients that you can malabsorb and how they will present, remembering that it's be rare that you get a patient presenting with every single one of these conditions. So if we malabsorb lipid, so fats, then we see a steatorrhea. It's a hard word to say. Steatorrhea. There's 2 R's to that one. Steatorrhea. That's like a really fatty, smelly, slimy stool. Very unpleasant. It's also what happens in giardia and giardiasis, so that's an important differential. So if you have someone presenting with steatorrhea, you need to probe and make sure that they haven't been traveling or they haven't been swimming in any freshwater lakes or anything like that. If you lose amino acids, you get a hypoalbuminemia. Albumin being the blood in the protein in the blood, and that results in edema. This will usually present as peripheral edema, so in the calves and ankles and things like that usually. Malabsorption of carbohydrates means you get that means that the carbohydrates enter into the bowel, and then you get bloating and flatulence. If you get low vitamin K, then you get poor synthesis of clotting factors. Specifically, it's factors 2, get my Roman numerals right, 7, 9, and 10. Gosh. 10. So the way you remember these ones, they're the ones that are also impacted by warfarin. It's the channels, if you're in Australia, the old TV channels used to be the ABC, Channel 7, Channel 9, Channel 10, before they moved to digital and got all the other random channels that are around. Anyway, poor synthesis of those clotting factors means that you then have bruising and you have a high INR. So that's like a poor clotting time. So it takes longer for your blood to clot. You get low iron. You can also get a low B12 and a low folate. However, you're more likely to present acutely with low iron than you are the B12 or the folate. So if we just kind of focus on iron at the moment, you're going to get a microcytic, hypochromic anemia. And so that will result in fatigue and pale conjunctiva. Because these patients tend to start to avoid high fiber foods, so they might not necessarily pick it up, but they start to realize, every time I eat bread, I just feel really sick, or every time I've been having salads and been but I had a Caesar salad once and it made me feel really off, and things like that. Or there's a dressing I know that makes me really sick, so I don't eat the salad anymore. All those kinds of things mean that they start to avoid high fiber food without realizing that they're avoiding it because of their celiac disease. That avoidance of high fiber foods leads to constipation as well. So often these patients have a really unfortunate scenario where they're constipated most of the time. They'll eat something to try and help with the constipation, and that will trigger a really severe bout of really severe diarrhea. Finally as well, because you've got blunting of your villi, so you have less surface area for... so this is all because you've got less surface area for digestion and absorption. You also get less of the sodium glucose transporter. Transporter. What that means is you're not really absorbing enough things in your gut, enough salt, enough glucose, which means you're not getting osmotic pull of water, so you get an osmotic diarrhea. And that's kind of compounded by all these other things as well. So I've said here as well that fatigue is related to iron, but also remember that the loss of all of these things, so if you're not getting lipids, amino acids, and carbohydrates in particular, then you're going to really be struggling to be getting in enough energy to live. So these people will be losing a lot of weight. They'll be really, really fatigued, really tired all the time, and really, really unwell, really. Cool. Alrighty. So we've already kind of talked a little bit about the... So we've spoken, one of the main reasons that you get the diarrhea and all these deficiencies is because you get that loss of surface area in which to absorb the nutrients. But it's also important to remember that the epithelium in the small intestine and the duodenum is not just there for absorption. It also plays a really key role in digestion. So you get loss of brush border disaccharases as well, which include things like isomaltase, sucrose, secrase, and a whole bunch of other ones activated by proteases. More importantly as well, you lose peptidases. So if you lose your peptidases, you get less activation of other enzymes. So you lose your aminopeptidases, carboxypeptidases, endopeptidases, and dipeptidases. They're all locked in the membrane. And so if you lose your membrane, you kind of lose those, as well as losing a whole bunch of your disaccharases and things like that. So it's a problem with digestion as well as just absorption. I did touch briefly on before how we kind of test for this. We do non-invasive testing by looking at screening the blood for those immunoglobulins. We can also do HLA haplotyping. So I spoke before DQ2 and DQ8 as being the most important. We find it's something ridiculous, like 99.9% of people with celiac disease will have at least one of these alleles. And so if we find a patient that's presenting with symptoms and they don't have one of these, then we're unlikely to go into very invasive testing until we've tried a few other things first. It might be something like Giardia, which is the other really key differential to consider when someone's presenting with these kinds of symptoms. If we want to go and confirm, if the patient tests positive for the antibodies and for the HLA, and we want to do a screening, we want to go past the screening to the diagnostic tests, we can do an intestinal biopsy, specifically of the descending part of the duodenum or the second part of the duodenum. We're looking there for that villus atrophy, the intraepithelial lymphocytosis, and crypt hyperplasia. All right, so that's our mechanism for today. It did get a little bit long and I got a little bit tied up in something else as we went along, so thank you for bearing with me. Hopefully that's been helpful, and keep an eye out. I'll try and put up a video soon going more into detail about this cross-presentation here process with the immune system. Thanks, guys.